everyone, and welcome to the University of Toronto Joint Center for Bioethics Seminar Series. I'm Angel Petropanagos, and I'll be the moderator for today's session. Our speaker today is Jeff Nisker, and his seminar is entitled, What's in a Name? Names Confusing Decision-Making in Reproductive Genetics. Before I introduce today's speaker, I'd like to let you know that the seminar is being recorded. This lecture, along with other archived lectures, can be accessed through the Joint Center for Bioethics website. The format of our seminar today is a presentation by our speaker, followed by a facilitated discussion period. We'd like to take a moment to acknowledge this land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and most recently, the Mississaugas of the Credit River. Today, this meeting place is still home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. We also stand in solidarity with the ongoing protests against racism and systemic discrimination. Now, it is my pleasure to introduce today's speaker. Jeff Nisker is a clinician, scientist, playwright, and author. Jeff's plays have toured 5 countries, and he has co-authored or authored over 180 peer-reviewed scientific publications, as well as many book chapters and books. When Canada fell behind other developed countries in public funding of BRCA mutation, testing for women at high risk, testing for women at high risk of breast cancer, Jeff's research morphed from basic clinical science research into ethics and health policy research. Jeff is a professor of Obstetrics and Gynaecology in the Schulich School of Medicine and Dentistry and scientist in the Children's Health Research Institute of Western University. Jeff has received many national research grants from Health Canada, Canadian Institutes of Health Research, Genome Canada, and other funders. Jeff has also received many national and international awards for his innovative research and education initiatives, including Canada's Royal Conservatory of Music's Excellence in Education Award for Public Engagement, Western University's Scholar Award for Innovation in Research and Education, and the Society of Obstetricians and Gynecologists of Canada's President's Award for the most significant contribution to the specialty. In 2010, Jeff became one of the first 2 in his specialty to be elected to the Canadian Academy of Health Sciences, and he has since received several other awards of excellence. Jeff was chosen by the Canadian Broadcasting Corporation as one of the 13 best minds of our time. Jeff, over to you. We're looking forward to your presentation.
Thank you so much. I really appreciate the introduction. It should be half as long, but thank you. What's in a name? Can I have the title slide, please? I don't have control of the slides, I'm afraid.
Can you see the slides there?
No.
Nope. Okay. Give me 1 sec. We'll try and pull it up again.
I did before the introduction, but I'll start talking so I don't waste the time. What's in a name? I mean, I'm talking about Juliet Capulet's claim. But in informed choice, names can be very confusing. And I contend they're no more confusing in decision-making than in reproductive genetics. Next slide, please. Now, I don't have conflicts of interest, but I have culpabilities. I've been part of this, as you'll see, and I give my mea culpa in advance. Next slide, please. I wish I could change the slides. So, what Juliet Capulet and Romeo Montague, their different names, created a conflict. I'd rather not, okay, just leave it there. Created a conflict that was unnecessary. Names were not important in Shakespeare's play. However, names are important in informed choice. The names we convey on reproductive genetic technologies, they are laden with substance. The name is important because I contend it shapes informed decision-making. Next slide, please. For example, this complicated slide, which I'll get to at the end again, this is a slide of supposedly mitochondrial replacement therapy, okay, where you are replacing the mitochondria in an oocyte. I'll use egg and oocyte interchangeably, to cure the woman's condition, so that she can have a very scare quote, healthy embryo fetus. But it's not mitochondrial replacement at all. What's going on here, you don't have a pointer, what's going on here is germ-line nuclear transfer. In other words, reproductive cloning. But nobody wants to call it that. Next slide, please. The reason that people in reproductive genetics don't want to call it reproductive cloning, because reproductive cloning is banned with criminal sanctions in every country that has legislation regarding reproductive genetics. Okay? Even the United States has a Human Cloning Prohibition Act, okay, and has had that for almost 20 years, okay? So we don't use the term reproductive cloning because it's illegal, but a rose by any other name may just not smell as sweet, okay? That's my thesis. Next slide, please. So what we're going to do, and I emphasize we're going to try to understand the importance of terms in informed decision-making. I'm going to use the example of reproductive genetics, but I'd like to think it involves all informed decision-making. Names are important for those who come to our care, absolutely. They're also important for REBs. Many of you sit on REBs or will sit on REBs. And, you know, when something comes to an REB, a name can influence your decision-making as to whether or not this research or this clinical procedure is permissible. It's also very important when laws are being made. Health policymakers and legislators are influenced by the names on the documents they see in front of them, and the names that are used by those who argue for the legality of whatever technology or procedure they're lobbying for. So it's very important to know and appreciate that words are charged particles, okay? It's especially important because genetic science is so complex, okay? And it's getting more complex all the time. And the technologies are getting more adventurous all the time. And it's also very important because we're not spending the time that we used to spend on ensuring an informed choice process. Everything's abbreviated. Everything comes up on sound bites and on graphs. So sometimes there isn't even a human being conducting the informed choice process. It's cheaper for someone just to press buttons on a laptop. So names are important, okay? Names are roses, but the technology isn't necessarily. May I have the next slide, please? So these are 10 names in reproductive genetics that I will allude to depending on how much time we have. They're roughly in the order with which they occurred over the past 30 years, but they're all in use today, big time. This is the nomenclature for reproductive genetics today. May I have the next slide, please? Preimplantation genetic diagnosis. So this is my assistant botching a biopsy of an embryo. I'm not blaming my assistant because I wouldn't even try to do this. She had golden hands. She could actually do this. But I'm not dignifying the procedure because I've been a big critic of this, because I don't want it to look like it goes smoothly all the time, because it doesn't. May I have the next slide, please? Slide before. This all started with Kary Mullis. It's okay, with Kary Mullis. He's an American who invented the polymerase chain reaction, and that allowed us to take one tiny little bit of DNA and amplify it up billions of times so that we can study the DNA in all kinds of circumstances. And as the Grateful Dead wrote, and then Fish and others have used, if you have a hammer, everything looks like a nail. Well, PCR was my hammer, although it wasn't the first. It was done in England first, London, the other London as we call it. May I have the next slide, please? So this is Alan Handyside, 1989, first bringing to the attention of scientists around the world the possibility of biopsying embryos. Okay, now he was looking for X-linked recessive conditions, and they could therefore determine if the embryo was a carrier of the X-linked recessive condition, because they could determine whether it was a male or female preembryo. Now notice I'm using the word preembryo. This was a name constructed in the very early days, like around 1989, of PGD research. It was felt that it would be easier to get the research and then the clinical applications through research ethics boards and hospital committees if they use the word preembryo rather than embryo. Again, what's in a name? Next slide, please. And of course, that's the name that we used. And in 1993, the Canadian Medical Association wrote all about our lab and what we had done. But I had already abandoned our lab because closed the doors, gave back a huge amount of grant money, because once the press got hold of what we were doing, calls came in from across Canada to come to our lab to biopsy embryos to avoid the terrible condition they were afraid their embryos would have. Now, if I was in an audience walking about you and sharing with you, I would look at you and ask, well, what sort of terrible genetic condition do you think that 58% of people who called my lab wanted to prevent? And many of you would put up your hands and say, being a woman. And that's the answer. They wanted to genetically select for males. Now, again, this was in the early 90s. I don't think it's the same today. At least I hope it wouldn't be the same today. May I have the next slide, please? Now, in 2012, a paper was actually written on the word preembryo. It was in Spanish. Could be Portuguese, I'm not sure, to be perfectly honest with you. And it was a very interesting paper talking about the history of the word and the fact that the word had never previously been used in animal or human embryology. The word was created in order for PGD to be permitted in research and in clinical use. Once PGD was accepted as clinical practice around the world, people stopped using the word preembryo. And again, I was guilty. I used the name preembryo in my REB submissions. Mostly it was for mouse, but when we were ready to do human embryo research in 1991, I used that as well. May I have the next slide, please? My lab, and you see on the wall, it doesn't say anything about human embryos. It says early pre-implantation cell screening, another Rose sort of name. What can be wrong with cell screening, except what we were doing was biopsying human embryos? You have the next slide, please. These were graphs we had on the wall talking about cell biopsy, cell screening. These were actually embryos, human embryos. May I have the next slide, please? Okay. Another Rose-like name is multi-pronucleate embryo that we used because if it didn't have the correct number of chromosomes, it wasn't human, right? We emphasized that these didn't have 46 chromosomes. However, it didn't take me long, and there are many reasons why I closed my lab and eventually did a PhD in ethics and health policy, was, there are many humans that don't have 46 chromosomes. They can have an extra chromosome, they can have a less chromosome. They're still humans in every way. They're still us. So why should there be an emphasis? Why should a multi-pronucleate embryo be easier to do research on than one with the regular nucleus? It doesn't make sense, but we didn't understand it at the time. Well, by 1992 we did. May I have the next? This name is still being used today, by the way. May I have the next slide, please? Anyway, so I closed my lab, gave back the money, and wrote this paper because I was really worried. I was really worried that I had contributed to opening a very scary Pandora's box. May I have the next slide, please? And I decided I really didn't know enough. So gradually trimmed back my clinical practice and applied to do a PhD in ethics and health policy at the Joint Center of Bioethics. May I have the next slide, please? The other thing I did, because I believe so much in public engagement, is I wrote a play called Orchids, which was first produced in the early 90s, and then went on across Canada and other country tour in around 2000. And it asks the question, what is normal? You have two women sitting side by side in a reproductive medicine waiting room. One, it has blocked tubes and just wants to have a baby, and the other one wants to have a specific type of baby, a baby that's very scare quote normal. And so next slide, please. So this play was produced and toured in various countries, and I learned a lot by the audience members' comments. Another name that I really hate is unsuitable embryo. And this is still being used today, okay? And that's because there are embryos that are cryopreserved that women choose never to use. They've completed their family. And when stem cell research became prominent, the stem cell scientists said, wow, there's all these embryos, all these human embryos, but they may not have looked exactly scare quotes, perfect under the microscope. And therefore, they thought they could be donated fresh to research. They felt that cryopreserved embryos wouldn't be as useful. Next slide, please. And then there's the healthy embryo. Now, we had a huge grant called Towards Single Embryo Transfer, big, huge CIHR grant in our department. I was just one of many investigators that we called the Healthy Embryo Grant. Well, I invited Roxanne McKittiuk, who's a legal scholar who works a lot with the Joint Center, to come to one of our meetings as I was building this ELS team, ethics, legal, and social team as part of this huge grant. Well, when Roxanne saw the first slide that said the Healthy Embryo Grant, she couldn't believe it. So this was a huge grant, multi-million dollars, that we were calling the Healthy Embryo Grant. And she really took umbrage with the use of the term healthy embryos. And we wound up writing a book chapter and editing a book that really looked at how conceptions of biomedical and social health of children impact conceptions of embryo health. And conversely, or not so conversely, how conceptions of embryo health construct new characterizations of the health of children, and for that matter, adults. This was huge. This was like a revelation that I take no credit for. Roxanne McKittiuk just saw it and said, "What are you guys doing?" Next slide, please. Here's the book that we edited with others. And Isabel Carpin, Carolyn McLeod, Francoise Bayless, Roxanne and I. May I have the next slide, please? Okay, so the next new kid on the block was the concept that you didn't need to biopsy embryos anymore. You could actually glean DNA from erythrocytes in the blood of a pregnant woman. This was fascinating. Diana Bianchi pioneered this work, and this was riveting. It was absolutely riveting. So we invited Deanna to grand rounds at our university, or university hospital, to tell us about this, because it would obviate the need for chorionic villus sampling or amniocentesis and other invasive procedures. But of course, I felt very eerie about this. Can I have the next slide, please? So Carol King, who was a very, very good resident, and at the time, who's now a faculty member of ours, and I use her picture with permission, talked about this. It was not then called, Diana Bianchi's term for this was non-invasive prenatal testing, because it didn't require chorionic villus sample or amniocentesis. So she gave a talk on this, and I, of course, went up to her very politely afterwards and said, "That's amazing, Carol. How do you know about this? I know this is in the research realm, but how do you know about this? You're a resident." And she said, "Dr. Nisker, you better come with me to the obstetrics unit." Next slide, please. So I walk into the obstetrics unit, and there is a stack, like a pyramid, of these Harmony test kits, a stack of them that a woman in our prenatal clinic could just pick up and take home and do genetic testing. I went to Shoppers Drug Mart, and they had a stack of these. So there's no genetic counseling, there's no informed choice process, you just swab your cheek, put it in to a test tube, and you send it in an envelope that's there to myriad laboratories in the United States. Sorry, it may not have been myriad this time, to a laboratory in the United States, and it comes back and tells you all about yourself. You know, I think now they can do 325 various genes. It was 150 when it first started. May I have the next? So I guess it's integrated genetics. I shouldn't have blamed Myriad for this one. All right. So I had a very good PhD student at the time, Meredith Van Stone, who couldn't believe this and did her PhD on this. And really looked at what were the ethics and policy considerations of non-invasive prenatal testing, which is what it was called, okay? There was a lot of ethical issues. First of all, it's not non-invasive. Sorry for the double negative. It is invasive. It's just a different type of invasiveness. You're getting all kinds of information without an informed choice process, let alone a full genetic counseling process. And it's not testing, it's screening. There is an enormous difference between testing and screening. Testing, you're looking specifically for a specific gene. Screening, you're screening the whole genome. It's very different. Well, as much of the genome as you can with the technology present. We have the next slide, please. And then this word donor embryo. So you see, you use a name like donor embryo for the embryo that a socioeconomically disadvantaged woman may give to a socioeconomically advantaged woman because they can make a lot of money doing this, but they're not truly donoring, they're selling. And this is huge in the United States. There are huge clinics that have, very scare quote, a stable, that's the term that was used, of embryo donors. They're basically oocyte donors, right? And then you take the male partner sperm and put them in the oocytes to create this donor embryo. So there's oocyte donors, sperm, usually from the partnership, that go in, and you create a donor embryo from donor, scare quotes, oocytes. Next slide, please. So we get back to mitochondrial replacement therapy, because this is basically the new kid on the block. And you're going to see a lot of this happening, where a couple wants to have the genetics of the male partner and the genetics of the woman partner incorporated into their child. It's not egg repair, it's not mitochondrial replacement therapy, it's DNA, germline, nuclear transfer, it's reproductive cloning. Next slide, please. We're almost there. I contend that this mitochondrial replacement therapy is the most deceiving, rose by any other name. And it was actually approved by HIFA, required a special dispensation from the anti-cloning legislation in Great Britain. They said they're not going to do it very many times, but they had been made a case and they did it that once and said right in advance, maybe once or twice a year, but that's it. But it's created a slippery slope. It always does. Next slide, please. Anyway, so we've been writing about it. Next slide. Now, the last name is this pre-implantation genetic testing. It's brand new. And I'm not going to go into it. But it puts a new spin on those of us who have criticized NIPT. It says, well, we actually can do testing under some circumstances. You can't keep criticizing us for just doing screening. And you can do this because of a new technology called next generation sequencing. And as new technologies develop, there will be more and more spins on the concerns that we had. And the concerns will be negated because a word is changed here and there. A name is changed to make it smell a bit sweeter. Next slide. I think this is my last. Next slide, please. Anyway, a group of voices that is denied, that we don't hear and we don't seek out to listen to as we talk about all manner of genetic screening, is the disability community. And I appreciated this when a woman who I wrote about in this book drove her chin-operated power chair into me and said, "Hey, doc, who writes stories, why don't you write one about the fucking way the system treats me?" And she taught me a great deal, and I'm forever grateful to her. But the system treated her in such a way that she died from an infected bed sore that was totally preventable if she would have had family and advocates in her behalf. And so, yeah, I call the woman Ruth, but that's not her name. But I owe you a lot, Ruth. Oh, you're no longer here with us. Okay, in summary, next slide. There is much in names in reproductive genetics, just as there are much in the motivations of those who create sweet-smelling names. And although many of the perfume names are no longer in a lot of use, many of the names still are. And that really does influence research ethics boards and clinical decision-making. Next slide, please. We are such stuff as names are made on. I'm using the word names instead of dreams, as Shakespeare used in The Tempest, and as Sam Spade, played by Humphrey Bogart, used in The Maltese Falcon. We are such stuff as names are made on. Next slide, please. I acknowledge the following persons for their help in research projects over the years. I am greatly indebted to them. Roxanne McKittiuk, Meredith Van Stone, Sue Cox, and many others. Next slide, please. These are my references, and thank you very much, and I'd be happy to entertain your questions.
Thank you so much, Jeff. That was really interesting, and I appreciate the deep dive into sort of the number of instances we have just in reproductive technology of where names have been softened or manipulated, and the opportunity now to think about a little bit more about how that might impact informed consent and the choices that we make. So I'm really excited for the discussion period. just first a reminder to our audience who are watching from YouTube. So if you'd like to ask a question, if you please type it into the YouTube chat function. And if you would like to ask a question anonymously or are not logged into YouTube, you can send your question via email to lori.bulchuk@utoronto.ca. And if you give me a second here, I'm going to go to our questions. Okay. So I don't see any questions there yet, but I do have a question for you. I have a few questions. I'm wondering if you could speak a bit more about what you see as the impact on the informed consent process. So I know with some examples you gave that there really wasn't that informed consent discussion. So maybe you could expand on the concern around the risks or the ethical considerations there, but also when there are informed consent discussions, but we're using these names in particular ways, what that might mean for consent itself.
So we have to understand that in the best case scenario, you have a genetic counselor conducting the informed choice process. In the best case scenario, genetic counselors are well trained. They're wonderful. They understand. They're usually very good communicators, better than we physicians. In the best case scenario, it is a genetic counselor who's going to conduct the informed choice process for genetics. However, genetic counselors can be influenced by names just like the rest of us. And more problematic is they can be influenced by the physicians for whom they are the gatekeepers. So for example, in private reproductive technology clinics, which are 99% of reproductive technology clinics, the last thing that the clinic owners are going to do is empower a genetic counselor who is scaring away their business. So there is this complex situation in reproductive genetics, a complex situation that I don't find in genetic counseling in regards to cancer treatment, because that's done within the provincial funded hospital oncology system. So it's just different from informed choice in cancer genetics and informed choice in reproductive genetics, both of which I've been involved with, and I just find it different.
So that's interesting. So it sounds like part of the complicating factor has to do with the privatization of the services and the sort of the maybe conflicts of interest when it comes to providing support to patients. And I'm wondering if you have any suggestions for strategies for people who are seeking to use reproductive technologies and entering into the system on how they can access information that is maybe less biased or access supports in addition or outside of the clinic that they're going to.
I hate to admit this, but I've been arguing for 30 years that physicians should never be business persons, that there is a difference, and that if it is health promotion or health care, it should be publicly funded no matter what. Otherwise, it shouldn't have the mantle of being health promotion and care. They're different, but again, it's what's in a name, right? If you have what's in initials, if you have MD after your name, people automatically think, well, there's no commercial stuff in it. After all, we live in a publicly funded health system. Not so for reproductive medicine. It's not publicly funded, but it's hard for people to say, wait a minute, this isn't publicly funded. So things are different than if this happened in a publicly funded situation. Although there are some units, ours, the unit at Mount Sinai in Toronto, and a couple others that still are publicly funded. But the vast majority are not.
Thank you.
I'm not criticizing the doctors who work there or the genetic counselors. It's just the name. It's just, and they're good people. And they follow the rules set out by the Canadian Fertility and Andrology Society, the Society of Obstetricians and Gynecologists. These are professionals who follow professional guidelines. So it's the professional guidelines that require scrutiny, and it's the funding. I believe if everything was publicly funded, things would be easier on everybody. Mm-hmm. And I think, you know, most physicians really enjoy publicly funding, not just to pay their salary, but so that they don't have to be business people. Mm-hmm. Thank you.
Thank you. So we have a question from Rebecca. They ask, based on your experiences, how can reproductive medicine be done in a more ethical way, considering that it's not going away and likely its use will be increasing? Reproductive medicine specific to reproductive genetics is the clarification there.
Oh, I think I answered that question while it was coming in and being translated, but just briefly, to be publicly funded. If we're going to use the word health in front of it, we're going to use the word genetics as part of it. I believe it should be publicly funded. I believe everything should be publicly funded. Maybe not augmentation, as was in my play Orchids. I'm not necessarily saying that augmentation should be publicly funded, but that's a whole different discussion. But certainly if we're going to speak in terms of health provision, health promotion, health care, publicly funded, I believe should be universal.
Thank you. So we have another question. Just wondering if you can comment on preclinical animal testing terminology and lay statements. And then they have a follow-up to say that REBs, with respect to REBs and public funding applications.
Yeah. I'm not big on animal research, to be perfectly honest with you. Yeah. So the Canadian Institutes of Health Research, who have been very kind to me over the years in funding not only my basic and clinical science research, but my ethics and health policy research with my co-investigators, has, sort of protocols. We have to do research on mice, maybe higher order rodents or lagomorphs, and I confess that my cancer research on endocrine, on estrogen basis of endometrial cancer was done in my old age home for elderly bunnies. We were kind to those bunnies and we let them live a long time, but we did keep them in cages, and we did do necropsies on their uteruses when they died. So, we just can't jump into human research. And I think research on primates is an abomination. I just can't buy it in any way, shape, or form. I just can't. It's abhorrent to me. So we have to find a way, and I think we will. I think we'll use not stem cell lines, but cancer cell lines, for example, cultures of cancer cells, and be able to test new therapeutic agents right on cells in a petri dish, rather than have to submit animals of any species, experimentation, it's sort of a Baha'i sort of thing. I'm not a Baha'i, but I have great respect for all religions. How we treat trees is how we treat animals. How we treat animals is how we treat humans. I think there's much wisdom in that.
I agree. Thank you. So our next question from Zoe, they ask if you think that the growing use of telehealth services during COVID will impact patients' accessibility to genetic counseling in the future, or maybe it has right now as well during COVID.
Well, it impacts the intimacy of being in a room with a person. I don't believe that telehealth can do that. In southwestern Ontario, which goes all the way up to Owen Sound, from Owen Sound to Windsor, our cancer unit, including our cancer geneticists, have used telehealth for 20 years as a way so people don't have to travel all the way down to London. Mm-hmm. Excuse me, but it's a definite second best. Being in the room, there's a certain intimacy. You look the person right in the eyes. There's a presence that you feel that I believe we lose, even on Zoom today.
So next we have a comment and a question together. So they say, thank you for this very interesting and informative talk, Dr. Nisker. They say, I truly hadn't thought about how these kinds of names related to reproductive genetics and maybe softened or changed for particular and concerning reasons. I was hoping that you could say a bit more about the disability community and any relevant experiences and or biases that may have occurred with your lab or your research. The story that you shared of Ruth, which we know is a pseudonym, was particularly impactful. So thank you.
So I would never dream of speaking for the disability community or for any persons with disabilities. I have had the great gift of persons with disabilities sharing their stories with me. I believe the Canadian Disability Society Conference is one of the best conferences I go to every year and highly recommend it. I just basically think of Ruth and how we let her down. And, I can write books, I can write plays, I can write poems about the experience, but I can never bring back Ruth, and our system let her down, and I'm part of the system. And call me Jeff, not Dr. Nisker.
Thank you. And so I think we have time for a couple more questions. But actually, first we have a comment from Sylvie, who says they suggest that we should add egg freezing and, quote, preserve fertility to your list. It is marketed in such a way that we do not discuss how success rates for actual successful embryo creation and pregnancy. And I can just second that, as fertility preservation is one of the things that I've worked on before. And I think there are a whole host of words and phrases and practices that are problematic and ethically concerning that I think could be added to your list.
No, thank you for that comment. And it's an enormous area because fertility preservation I have participated in as a clinician starting 30 years ago for cancer patients. But as you go to fertility preservation for social purposes, such as, career and other reasons, and I think we're really talking about oocyte preservation frequently now. Um, it's something for discussion. I believe it's up to the woman to decide these things. I'm very worried about taking empowerment away from women in the name of ethics and health policy. I think a woman wants to delay her childbearing to a time where her age would make it much more difficult, then her choice. And I see no problems with that. It's the woman's choice.
Thank you. So in thinking about so what I know about fertility preservation and the sort of the language that's used in that context, so I have a question about sort of the role of media and social media in particular. So I know your talk really focused on thinking about the lab context and the clinical context and informed consent and how the words that are used in that context matter. But I've seen in lots of ways, the language that we take from the clinic, that we take from the research, has sort of permeated into the public sphere. In fertility preservation, it's in ads to sort of freeze your eggs. Then there are social media groups like on Facebook where people have used or co-opted different terms that might be problematic or might be advantageous for them in particular ways. So I'm wondering if you have any thoughts around sort of what it means when that language goes outside of the clinic and it's in the sort of the public conscious, what that might mean for us.
So there's a difference between cryopreservation of sperm, where a man, before he undergoes cancer treatment, ejaculates, the sperm is collected, spun down, and frozen, and a woman undergoing a surgical procedure to cryopreserve her eggs. The latter requires a very lengthy and informed choice process. But ultimately, the woman should have the choice. Again, I'm more intimately involved with women undergoing cancer treatment, but because I really haven't practiced reproductive medicine for many, many years, but the people who practice reproductive medicine today have similar feelings. And I don't believe we can take away informed choice. No matter what the media does, it just makes us, gives us the additional requirement to spend more time in an informed choice process. But it's ultimately up to the woman to decide these things, and up to professional organizations such as Canadian Fertility and Embryology Society, Gynaecology of Canada, Canadian Bioethics Society, to make sure that the choices a woman has are portrayed in a fair, risk-conscious manner, but ultimately empower her with the decision-making.
Thank you. Okay, so that makes sense. So if, in the public discourse, we might not really understand what those technologies or those reproductive options involve. And it's really up to the clinicians to make sure that that informed consent process fills all those gaps that we might have.
Yes, we include genetic counselors.
Okay.
Physicians are not that good at doing this. I think we're getting better, but it's ultimately with the participation of a genetic counselor, in my opinion, that reproductive genetics and a real true informed choice process can occur in reproductive genetics.
Thank you. So we have, I think we're running up on 5 o'clock, so we have time for one more question. And Wade asks, where can I get a copy of Orchids? More now than ever, art needs to be a conduit for so much of the pain, treatment, collaboration, and normalization of all the things you've spoken about today. And incredible talk. Thank you.
Thank you. It should be available in chapters and other bookstores. The publisher is Iguana Press. And what I will do is I will contact the publisher just in case you can't order it for some reason through chapters or Kohl's. I shouldn't just say or Indigo, or I will circulate ways that you can get your hands on all my books that deal with reproductive genetics.
That would be great. So maybe we can invite those who are interested, and if they have any difficulty accessing the book, to reach out to Lori Belchuk, and she can connect you with Jeff, and we'll make sure you can get a copy. So unfortunately, it's time to draw today's seminar to a close. So I just have a couple of announcements to make before we thank our speaker. Our next seminar will be next week on Wednesday, February 3rd. Andrea Bianchi, Catherine Stanley, and Callum Sutander will be discussing the ethical defensibility of harm reduction and eating disorders. To sign up and receive our weekly seminar reminders via email, please send an email to jcb.info@utoronto.ca. For those of you who are CSB students enrolled in the CSB Student Seminar course, please remember to keep track of your attendance. And finally, Jeff, on behalf of all of us today, thank you very much for your presentation. It was really insightful and has given us a lot to think about. So thank you.
Thank you for inviting me.